Okay, so we'll be discussing about the bacteriology. So, bacteriology is actually part of your microbiology. So, microbiology is divided with your parasitology, with your bacteriology, and we have also here your study of your fungi, mycology, and you have also your virology for the viruses. So we have already we're done already with the parasitology. So for this STEM, we'll be dealing here with bacteriology. So for this subject, okay, so we'll be discussing here about all the bacteria. So medyo mas broad ito compared with your parasitology. Um, for the prelim, we'll be discussing all about the introduction regarding the bacteriology. For the midterm and finals, we'll be discussing the individual bacteria with their different morphology, with their characteristics, with their biochemical reactions, and the different diseases associated with each of the bacteria. Okay, for this subject, we'll be using this book. Okay, this is your Diagnostic Microbiology by Mayhon. Okay, this is the fifth ed. You could use also this book here for your virology and your mycology. Okay, so again, when speak about your bacteriology, so this is part of your microbiology. So what we are dealing here with the microbiology are microorganisms. So we speak about the microorganisms, so those are very small organisms wherein they could only be seen, identified here by the microscope. They could not be seen by using our naked eye. So we just utilize here the microscope for us then to visualize those very small organisms. So we start the discussion with the subject with historical milestones with the development of the bacteriology, the microbiology as field of your medical science. So we start here with, okay, so scientists before believe here that a particular disease is caused here by a specific causative agent. So they are thinking that a specific disease is being caused here by a causative agent that may be in the form bacteria, and we have here other organisms. So we have here Lucretius believe that a specific disease again is caused here by a specific agent or a microorganisms. Fracastoro, on the other hand, associate here the syphilis transmitted primarily here by Seminaria morbi, or they call it once the bad seed. Seminaria, so currently naman, your syphilis is a sexually transmitted disease. It's caused here by the bacteria um, Treponema pallidum. This one is a spirochete na bacteria. And it's being transmitted here by sexual. So this one is by your seminal fluid. Although it's being also transmitted not only by your sexual contact, but as well as it's also transmitted here by blood transfusion. So basically, this one is correct with the assumption that the syphilis is caused here by the bad seed or the Seminaria morbi, Fracastoro. The next one, we have here Stiluti. Okay, Stiluti here able to identify or the presence of the organisms here on their structural level, cellular level with the use of the microscope. But Stiluti here uses a microscope supplied by Galileo, not for identification of the microorganisms, but it's been used, he tried to use the microscope here for identification of bees and weevils. Okay, the next one we have here Robert Hooke. Okay, Robert Hooke here first to develop the compound microscope where he tried to identify the presence of the little compartment, or he tried to describe that one as the little boxes. He tried to use here the bark of the tree. So, or okay, the cork or the bark of the tree for him to identify the presence of those little boxes under the microscope. So I tried to examine the bark of the tree. We're able to identify the presence of those little boxes or little compartment. Actually, those little boxes or little compartments are actually mga individual cell on the bark of the tree. And that forms actually the start of the cell theory. So believing here that living organisms are actually made up of the basic functional unit cell. But so the cell here try to group and eventually become tissue, and the tissue become try to group here to form an organ, and organ become here the organ system. So this forms part here very important discovery in the biology as the basic principle here, um, confounding the basic principle of the cell biology because of that. Okay, the next one we have here Anton van Leeuwenhoek. So Anton van Leeuwenhoek here describes the microorganisms under the microscope, and it try to describe those microorganisms here as beasties for animalcules or animal-like organisms. So Anton van Leeuwenhoek here try to identify the use of his microscope the different microorganisms, the existence of those microorganisms here by using the rainwater as a sample, his own feces, or even his own urine, and scrapings from his teeth to identify those microorganisms. And therefore, Anton van Leeuwenhoek here was then considered as the father of the protozoology. Okay, the next one, we have here the theories of the origin of the living organisms. So we have here the three contrasting ideology as to where the living organism came from. So the first one, we have here spontaneous generation. So spontaneous generation advocates actually believe here that the living organisms arises from a non-living organisms. So non-living like mga organic matter, and they call that non-living organism here as the origin of the living organisms as a vital force responsible here for the production or the formation of a living organism. So sa biogenesis naman, they try to believe here that the living organisms really came from a coming from here a pre-existing living organisms. Okay, then we have here the theory of the spontaneous generation. So again, the advocate of the spontaneous generation believing here that the living organism arises from non-living organism. So one of the advocate of that we have Aristotle believing here that animals came from soil. Then we have also here Francisco Redi try to demonstrate here the growth of the maggot from the decaying meat. They try to make an experiment here. They call it was the 3 jars experiment with the decaying meat on that. So we have here the 3 jar we being set up. The first jar will have here the decaying meat inside. Then we have here the paper covered here, or the opening here is being covered or sealed by the paper. And we have also here the second jar here is left open with the decaying meat here. Third one, you have your decaying meat. This one is being covered here by a gauze spot. Okay, so after several days, so what are you expecting here? So this one, okay, the decaying meat here, since this one is fully covered here by the seal here, so walang nag-grow na maggot on this one. And this one, since open siya, the maggot here, nag-grow siya dito on the decaying meat. And the third one, your maggot here able to grow, only that the maggot here dito sila sa taas. In your cover here, not on the decaying meat itself. So sabi ni Francisco Reddy here, so the maggot daw nag-grow siya from the decaying meat. And decaying meat here is considered as the vital force, which is non-living thing. But then if you try to analyze the experiment, so ito, okay, wala ito, hindi mag-grow ang maggot dito because eventually, hindi makapasok ang fly. Di ba your maggot, It's actually coming from the flies. Okay, so hindi mag-grow ang maggot dito since naka-fully sealed ito and therefore hindi makapunta ang fly directly to your decaying meat and therefore hindi sila makapag-lay ng egg and walang maggot. So this one, what are you expecting here is since na may decaying meat ka dito and open naman siya, so the flies will be attracted on this one and the fly will going to feed on your decaying meat directly. And then eventually they try to lay their nests, lay eggs on the decaying meat, and eventually after a day, several days, magkakaroon na siya ng it will lay maggots na siya. And this one, the third one, this one could able to lay maggots pa din, only that the maggots here dito sila sa taas ng cover. Why magkakaroon ng maggot dito? Because remember, you just have covered this one with gauze pad lang. And this one, still naamoy pa din ng flies natin na may decaying meat dito. And eventually, so since attracted sila dito, the fly here will just go in this one, concentrate on this area kasi hindi sila makapasok. And also, that become their nesting place. So, dito sila maglilay ng egg. Okay, so akala sabi ni Francisco Ready, so believing here that the maggot daw came from the decaying meat, but actually it's not. Okay, but it came from the flies na naglay ng egg because they are attracted to your decaying meat. But then again, Francisco here is an advocate believing on the spontaneous generation. Okay, the next one we have here, John Needham. So si John Needham tried to make an experiment, okay, supporting the theory of your spontaneous generation. Try to made a setup here with your flask containing the nutrient broth here, and then try to heat that one. So ang ginawa niya, meron siyang nutrient broth dito, and then try to heat this one. Okay, after heating, he tried to cover this one. Okay, and after several days, nakita niya na naging turbid ang nutrient broth natin. Nutrient broth niya, that would signify na may nag-grow na organism or bacteria on that. So sabi niya, okay, nagkaroon ng bacterial growth dito is because na may vital force na ito. Okay, may vital force na daw ang nutrient broth, that's why nag-grow ang bacteria. So, still advocating spontaneous generation. Okay, but then again, another priest here. Actually, this is a priest. This is another priest. So, Spallanzani here try to reverse the findings of Needham. So, ginawa niya, although try to modify the experiment made here by Needham. So, ginawa naman ni Spallanzani. So, still have here nutrient broth. Then, ang ginawa niya lang kaso, try to put on nutrient broth here. And then, kinover niya agad bago siya nag-heat. Okay, so, after heating process, walang nag-grow na bacteria, walang turbidity or walang growth dito. So, what's the meaning of that? Okay, so, wala nag-grow dito is because hindi nakapasok ang air. And eventually, the air itself contains the microorganisms. So, since na bago ka nag-heat, okay, di ba, to try to seal that one with a cover and heat that one. So, eventually, hindi na makapasok ang air natin. Kung may nakapasok man dito kanina, or like, if you have been contaminated, like, your nutrient broth here has contaminated na siya bago pa lang, but then, ito sa Speranza, kinover niya na agad dito, try to heat, mamamatay na siya after heating process. Therefore, after that, heating here, wala siya, or wala nag-grow na bacteria on that. Ito kay Needham naman, nagkaroon ng turbidity here because although na-heat niya, pero open pa din kasi ito. Nag-cover lang siya after heating process. So, from the time na naka-open ito, eventually the air enters this one, and therefore, the microorganisms accumulates. Okay, and then, they try to grow here. So, sabi ni Speranzani, it's actually not the vital force in the nutrient broth here that's responsible for having a new organism that form the microorganism growth, but rather, sabi ng Spallanzani, the air itself na pumasok contains the microorganisms. So, ito ay try to advocate on the spontaneous generation. Spallanzani try to advocate on the biogenesis. Okay, then we have also here the biogenesis, so another theory as to the existence or the formation of your living organisms. So the advocates of the biogenesis believing here that the living organisms really arises from pre-existing living organisms. Dapat living din ang pinagmulan niya. So this one has been proposed here by Rudolf Virchow, which is advocating here that your living organism here is the one responsible for the production of another living organisms. Another one, we have here Louis Pasteur tried to demonstrate here several experiments proving that the microorganisms are present in the air. Kaya nga nagkaroon ng growth dun sa ating sa experiment ni Needham and Spallanzani is because again the air enters the setup. In the case of your kay Needham, nag-enter ang air sa kanyang flask, containing here the nutrient broth, and that one, the air contains the bacteria, and that's why nagkakaroon ng bacterial growth on that. And then Louis Pasteur really believe here, advocates that the air itself contains the microorganisms. Pero ang air hindi siya nag-create ng microorganisms. Pero ang air may microorganisms. Okay, therefore, in order for us then to make our environment, like for example, your setup sterile, dapat it should be prevented here from contamination from the external factors, including the air, and become here that serve as a basis for your aseptic technique. That's why pag nag-culture tayo, make sure na gumagawa tayo ng aseptic technique by heating that one, including your your Petri dish and all, para hindi siya makontaminate ng other organisms. And therefore, Louis Pasteur was then considered to be the father of the bacteriology. So, Louis Pasteur here tried to prove that the air contains a microorganism through his swan neck flask experiment. So, from the setup here, so it's a flask wherein the neck of that has been bent para magiging swan neck siya. So, from this experimental setup here, so we have here the sterile solution here. Then, meron tayong naka-bent na neck or side arm ng ating flask ni Louis Pasteur. And try to heat this one. So, after heating process, okay, so basically ang air makapasok dito because hindi naman siya fully na close ito. But then again, ang microorganism here in the air, although ang air mo nakapasok, pero ang microorganism in the air, hindi siya makapasok because they are being trapped here in the side arm or kung nakabent ang arm ng ating flask. And therefore, the microorganism here being trapped on this part, and the air could enter, but then again, the air has already been free from the microorganisms. And therefore, sterile solution here still remains sterile kasi hindi siya, although nakapasok ang air, but ang microorganism the air, hindi siya nakapasok, and therefore, they are then prevented here from contaminating your sterile solution, and that's become the basis of the aseptic technique. So, correct siya in terms na sinasabi niya na the air contains the microorganisms, and then the microorganism itself or the air itself do not create the microorganisms. Okay, so Louis Pasteur, also known here for his pasteurization. Pasteurization of process whereby you try to subject like your milk, for example, into high temperature. That's to kill the bacteria on that. Another one, fermentation process whereby is a wine-making process. So you are heating the grapes. For example, here, try to heat that wine at temperature enough to kill the microorganism, but not enough to kill the yeast. The yeast is responsible here for the fermentation process. And that's pasteurization fermentation is also attributed to Louis Pasteur.